Good morning. Today in the context with your animal physiology, we will see movement and locomotion. Right? Now, in this first I'll tell you what is locomotion. So, what is locomotion? You know, this animal, they have a significant feature of moving from one place to another place, right? And this living organism, they are having the significant feature of this locomotion. So animal and plants, they exhibit actually wide range of movement, right? You remember this locomotion when we say, when there is change in the position, right? When there is change in the position. Now, next I'll tell you about the movement. This movement, if you see, there is little bit difference in case of plants and in case of animals. In plants, what happens? This may be either due to growth, right? So when there is growth in the plants, these changes are made, right? Or second, there may be changes or internal body components with respect to the one another. What I said, this in plants the movement is shown in respect to the growth and also in change that is made in the changes made in the internal body parts or body compartments with respect to one another. Right. So the growth may be, this movement in plants may be due to growth or the changes that is made in the internal body compartment with respect to one another. So depending upon this, we can divide this movement as growth movement and this is non-growth movement. The example in growth movement, we can give the example of germination of seed. You know, when there is germination of seed, there is development of plumule and radicle, isn't it? So when there is development of this plumule, this moves upwards. So there is movement towards upside, and radicals, this moves downwards. Okay, and then in non-growth movement, this usually occurs the movement in response to environment. In response to environment. Movement in response to environment, right? In response to environment, you know, when you might have seen when there is growth of the movement, that is all. This plant always grows towards light. So there is photonasty, then hydronasty, epinasty. This all type of nastic and tropic movements. This you have learnt in class 10th, right? Now next, let's see the movements in animals. This movements in animals, this also differs in case of your unicellular organism and multicellular or higher developed animals. Right. So in unicellular organism, what happens, like you see in case of amoeba, paradisium, right? Here the movement is very simpler. The movement is simpler, and this movement is mainly due to movement in the protoplasm. That is due to the streaming movement of the protoplasm. That is because of streaming movement of protoplasm. Because of, right? Now, if you see the example of this movement in unicellular organism, there is movement of flagella, movement of cilia, movement of tentacles, isn't it? So, the example will be movement of cilia, flagella and tentacles. Right? Now, let's see the movement in multicellular organism. Here in the higher animals, there is movement of the limbs or eyelids or jaws or tongue, etc. Right? So, when in case of multicellular organism, you will see there is well developed movement, right? And this movement is also, this can be the specific part, that is movement of the limbs, movement of the tongue, movement of the head, movement of the eyebrows, movement of jaws, right? So this movement in higher animals is very specific. Now this, when I told you that when this movement, this is result in change of location, when the movement that result in change of location, that movement is known as locomotion, right? So this locomotory movement can be your swimming, walking, running, everything, right? Now the locomotory organs, they actually need to be very, very different from those which affects the other movement. What I said, the locomotory organs, they need to be very different from those which are affecting the other movements, like in paramecium. In paramecium, what happens, you see? The cilia, this helps in the movement of food through the cytopharynx, as well as also help in the movement of locomotion. But in this higher animals, they are different, right? In unicellular organisms, these locomotory organs, they purpose many works, like your they do not have a specific locomotory organ, right? But which helps in them, that is cilia. So this cilia, which helps in the movement of the food through the cytopharynx, this also helps in their movement of locomotion. In case of hydra, you know this, they have tentacles which actually catches the prey, as well as this helps in the locomotion, right? So remember that all movement are not locomotion. All movement are not locomotion. Please remember this point. All movements are not locomotion, but all locomotions are movement, right? I think you have understood this point, right? Why all movement are not locomotion? Because when you see this movement that is occurring in the plants, they are not locomotion, right? When there is a change in response to the, that is your light, or this growth movement or non-growth movement, they cannot be the locomotion, right? Now, in higher animals, what will you see? Whatever the cause of movement, that may be either in search of food, or in search of shelter, or search of mate, right? Then in search of suitable breeding period, then favorable climatic conditions, or to escape from the enemies. These are mainly the some of the causes that I told you, right? Now let's see what are the types of movement. You will see what are the types of movement. This movement can be amoeboid, movement, ciliary movement, amoeboid movement, ciliary movement, and the muscular movement. This I am telling you about the animals, right? So let's first see the amoeboid movement. Amoeboid movement. This I told you amoeba is a unicellular organism, right? And the movement here is of simpler form of movement, right? And this is mainly because of the streaming movement of protoplasm and cytoskeleton. Cytoskeleton is formed by microfilaments, etc., isn't it? So here this movement is because of streaming movement of protoplasm, and this involves the cytoskeleton, that is your microfilaments, microtubules, etc., right? Then second, in this type of movement you will find in the amoeba, WBC, that is leukocytes and the macrophages. Yes. So this amoeboid movement are found in amoeba, macrophages, leukocytes that are present in the blood, right? White blood cells. So if you have to write the movement in the amoeboid movement in blood cells, then you will write the leukocytes, macrophages, etc., right? Now second is the ciliary movement. Second type of movement is the ciliary movement. This ciliary movement, this is actually occurs in the tubular organs. There are tubular organs which are lined by the ciliated epithelium, and they show very coordinated movement of the cilia, right? Like you take the example of your trachea. This trachea is lined by coordinated epithelium, and this helps to take out the debris, dust or foreign particles, whatever is inhaled, they are trapped by this mucus of this mucosa, and then they are moved outside, or they are thrown outside with the help of this ciliary movement, right? Then second is the passing of ova from the female reproductive duct, right? So these are some of the movement which are example of the ciliary movement. So this occurs by cilia, right? And here the examples I had already told you, that is the movement that we find in the trachea and the reproductive duct of the female during the passage of ova, right? Now this third type of movement I told you is the muscular movement. Muscular movements. This muscular movement, this mainly example you will find the movement of the limbs, jaws, tongues, etc., right? Here you remember that this muscular movement is the cause of actually a very contractile property of this muscle. Contractile property of the muscle, this actually helps in the muscular movement. So this also needs proper coordination of this muscular, skeletal and the nervous system. Remember, there is need of coordination between muscular, skeletal and nervous system. This is very important point. So this muscular movement, this actually requires very much coordination of the muscular, skeletal and nervous system, right? Now this we will see in detail, right? Now first of all, let me tell you about the muscular system. See, the muscular system, you know, this muscles in our body are of three types, right? Skeletal muscle, cardiac muscle and the smooth muscle, right? So the skeletal muscle, they are also called striated muscles or voluntary muscles, right? Now we will see first this skeletal muscle, right? And why they are called skeletal muscle? Because they are attached to the bones, right? So here remember that this skeletal muscle is mainly made up of many muscle bundles or the physicals which actually held together by a common collagenous connective tissue, and this then we call fascia, right? This common collagenous connective tissue layer is called fascia. So what I said, the muscle is made up of your many muscle bundles, right? Many muscle bundles, and they are held together by fascia, right? Then each muscle bundle, this has many muscle fibers. This is made up of many muscle fibers, right? And each muscle fiber is covered by sarcolemma. This is a enclosing plasma membrane, right? And this sarcolemma actually encloses the protoplasm that we call sarcoplasm, right? Now this muscle fiber, you know, this is actually syncytial. What I am saying? Syncytial. Muscle fiber is syncytial. That means here there are many nuclei. So the sarcoplasm contains many nuclei, right? So the muscle fiber is syncytial, right? Now we will see the arrangement of this fibers. You know, this sarcoplasm, this has actually many parallelly arranged filaments. Parallel arranged filaments are found in the sarcoplasm and they are called myofibrils. This contains many myofibrils or myofilament, right? So this contains many myofibrils or myofilament, which are actually the parallel arranged filaments. And the sarcoplasm, this also has the sarcoplasmic reticulum, which is actually the source of calcium ion, or here the calcium ion is stored, right? So the sarcoplasm has many myofibrils or myofilaments and sarcoplasmic reticulum, which contains the calcium ion. So remember this structure. So sarcoplasmic reticulum is the storehouse of calcium ion, right? Now, why this is called striated muscle? Why this is called striated muscle? Because this has alternate light and dark bands. So this appears striated, right? So this appears striated. That is why this is known as your striated muscle. Now this stria is actually because of two types of protein that it contains, right? This is due to the actin protein and myosin protein. So what I said, there are two types of protein, actin and myosin protein presence, right? And there is alternate dark and light bands on which due to which this striated appearance is seen, right? Now this alternate light and dark protein, I told you, is mainly due to actin and myosin. So the light band, as you see here, I had drawn this light band is due to actin protein, and this is known as your I band. This is called I band or isotropic band. Isotropic band. But this myosin, which is dark band, actually it is thick. So this forms A band, that is an isotropic band. Okay, this forms the anisotropic band. Okay, now this isotropic and anisotropic band is clear. What I said, that there is light band that is made up of thin protein actin, right? And there is a thick band that is of myosin, and this is due to the myosin protein, right? And this thick is known as your A band or anisotropic band, right? Now here you can see that these bands are arranged parallel to each other, right? And parallel to the longitudinal axis of the myofibril. Remember, these fibers, these bands, they are arranged parallel to each other as well as parallel to the longitudinal axis of the myofibril, right? Now this actin filaments, as you have seen, that they are thinner than the myosin filament, so we get thick and thin filament simultaneously. This you see here, right? And there is presence of Z line. There is presence of Z line, right? Between this I filaments. Right? So this is your I band. Here also you will find the same. Right? So there is a Z line between the I band which actually bisects it. So the I band is actually, you have seen, they are firmly attached to this Z line, and the thick filament in this middle that is also this held together to what I said, this thick filaments in this middle, they are also held together with the help of a thin filamentous membrane that is called M band. So remember, this in the A band there is a thin filamentous membrane that is called M band, and with the help with this M band, this myosin filaments are attached or they are held together, right? And here in the I band there is in between Z line. So one more thing you remember, the portion of myofilament that is lying between these two Z lines is known as sarcomere. Sarcomere. And this sarcomere is actually the functional unit of muscle. Functional unit of muscle. So sarcomere is the functional unit of muscles, right? Now A and I bands, they are arranged alternately throughout the length of the myofilament, right? So in case, first we will see the resting phase or the resting stage, right? So in resting stage, here you see the edge of thin filaments, they are present on either side of the thick filament, that is myosin filament, right? And they are leaving a center, right? A central part of thick filament. What I said, this is the myosin filament and these are the actin filament. In resting stage, the edge of this thin filament actin are on the either side of thick filament, and here you can see that the free ends of the thick filament, this is not overlapped, right? Now, so there is a living central part of the thick filament, isn't it? This thick filament part is in this thick filament central part is remaining, right? Now this central part of the thick filament, which is not overlapped by the thin filament, is known as H zone. This is known as H zone, right? In resting stage, the portion which is of the thick filament which is not overlapped by the thin filament is known as H zone, right? This part, you can see this is the part of the actin filament which is not overlapped by this myosin, right? So this is H zone, right? This you will get confused with this Z line, right? So I am showing it here, okay? I am showing it here. This you can see, this is the part of the actin filament which is not overlapped by this myosin. So in resting stage, this is called H zone, right? Now the central part of this thick filament which is actually not overlapped by this thin filament is called H zone. Each actin filament is made up of two F actin which are actually helically wound to each other, right? And you know this F actin is actually the monomer of G actins. G actins, that is the globular protein. There are two helically bound F actins, right? These are the two helically bound F actins. And these two filaments, the two filaments of tropomyosin, they also run along the length of this. There are two filaments of tropomyosin which also runs along this length. So this is two filament of F actin, which is the monomer of the G actin or globular, and this is two filament of tropomyosin, right? Tropomyosin, which is also helically bound, right? And they run along the length of this myofilament. And this is very close to the F actin complex protein, right? Now, this there is one special type of complex protein that is called troponin. Troponin, and this is also present or scattered at some places of this tropomyosin. So this is troponin, right? Troponin. This troponin is also distributed at some places of tropomyosin. Now, in resting stage, what happens? This troponin subunit, this masks the active site for the myosin on actin filament, right? And this myosin filament is also, this is a polymerized protein, and there are many monomeric proteins are found in myosin. They are called meromyosin. Meromyosin. So this myosin filament is polymerized protein made up of many monomeric proteins. They are known as meromyosin, right? And this meromyosin actually constitute one thick filament, right? Now, this meromyosin, this has two parts. The globular head, this has two parts. Globular head with the short arm, and the second part is the tail, right? Now, this globular head with the short arm is known as heavy meromyosin, that is HMM. HMM, heavy meromyosin, and this is known as light meromyosin, right? Now, this head and short arm, they usually project outwards at the regular distance, and the angle that they form with the meromyosin, and this is known as actually crossband, right? What I said, that there are two parts, right? Globular head and the globular, this is head with short arm and the tail, right? Okay. And they remain projected outside and this contains the your globular head and this is your tail, right? Now, this globular head is actually an active ATPase enzyme. Active ATPase enzyme, and this has the binding site for ATP and also active site for the actin. This, remember, this globular head is actually ATPase enzyme, and this has two binding sites. This has two binding sites, one for the actin and the another for the ATP, right? ATP. That is, here the ATP, ADP and IP, they will form ATP, and when required energy is required, this ATPase will cause hydrolysis of this ATP and convert this ADP plus inorganic phosphate, right? So you see, this myosin is made up of functional monomer, that is the monomeric protein meromyosin, and each meromyosin has one globular head. This is the globular head with short arm and the tail, right? And this head, this has the two things, that is the actin binding site and the ATP binding site, and this is actually enzyme ATPase. Right. Now I told you that this forms the cross link, right? The bridges, the cross bridges or cross link, and they remain protruded outside. So when, suppose this is the and there are fibers, so when there is contraction, they make the cross bridges, right? Now let's see the mechanism of the muscle contraction. The best theory this explains the mechanism of muscle contraction is the sliding filament theory. By the sliding filament theory, I am rubbing this portion. Sliding filament theory. So here actually what happens, there see, the contraction of the muscle fiber which takes place is actually by sliding movement of the thin filament over the thick. There is sliding movement of the thin filament over this thick myosin filament. Right? Now, contraction of the muscle, this is mainly initiated by the signal which is received from the CNS via motor neurons. Now, this motor neuron along with this muscle fiber, when they are attached to this, forms the motor unit. What I said, the motor neuron along with the muscle fiber is forming the unit, right? Now, you see, there is different different steps in this contraction, right? So what happens first? See, this is the resting stage. This is the resting stage, and in this resting stage, you are seeing that there is a H zone, which is actually the non-overlapped part of actin protein, which is not overlapped over the myosin, right? Now, when the contraction occurs, actually first I will tell you the method, then I will show you by the steps, right? So when this contraction occurs, this actin fiber, this slides over the myosin, I told you. So what will happen? The distance of this sarcomere will be reduced and the edges will be almost disappear. But when there is more maximum contraction, then what will happen? See, maximum, when there is maximum contraction. So this is the first stage, right? And in second stage, what will happen? This will come, isn't it? There is no H zone, right? And when there is maximum contraction, what will happen? This will overlap over this myosin. This will be the stage of your maximum contraction. Means you see the Z, the distance between two Z or the sarcomere is reducing. Here the sarcomere is very high, this is less than that, and it is least. Right? So this is the maximum stage of maximum contraction. And there are forms, the presence of this cross bridges which are formed by, I told you, the globular head portion of this meromyosin. Right? This actually is attached here. In another diagram, I will show you the next method that will be more clear. Right? So this is clear. So here this Z, there is this diagram is mainly showing you that there is overlapping of this actin filament over the myosin. Right? This is progressive. Right? Here in initial stage, resting stage, there is a space where no actin filaments are overlapped over myosin. But here you can see this has almost overlapped, this actin filament has overlapped the myosin. And in this you can see that there is so much contraction or maximum contraction, then this actin filaments also overlap this. Right? So is it clear? Now here I will show you how this troponin, tropomyosin, they actually take part in this muscle contraction, right? Now initially you see if you see I am showing you the actin and the myosin filament, right? So this myosin filament, you know, this is made up of viromycin, right? Now this is the myosin filament, right? Then now this myosin filament is having one globular and short head, isn't it? And this is the top actin, that is actually the actin filament, right? Let me first draw this diagram. Now this is, as I told you, that this has the globular heads, which is having the ATPase enzyme, right? So this is the resting stage. Now this is your actin filament, this is myosin filament, and this is your ATP binding site and the actin binding site of the myosin, right? Now when there is stimulus comes, right? So here what happens, you know, suppose this is a skeleton and this is a muscle, right? So this is the myoneural junction I am showing you here. And this, what happens when the impulse comes, when this impulse comes through this nerve axon, then here there are some vesicles which release the acetylcholine in this gap, that is the synapse or the myoneural junction, right? And this acetylcholine, which is then causes the activation of the action potential, that is, it opens the sodium potassium pump and there occurs transmission of the impulse, right? So the muscle contracts. Now this actually forms here the cross bridge. You see, this is the head and this is forming the cross bridge. This is forming the cross bridge, right? So this is the step showing the formation of cross bridge, right? Now then there is sliding or rotation. Then there occurs sliding or rotation. You see this? This is sliding, right? So this is showing your sliding or rotation movement, right? Now here the inorganic phosphate and ADP is formed and this ATP is broken down, right? Now the next step, there occurs breakage of this cross bridge, right? there occurs breakage of cross bridge, means it comes to the normal resting stage. This is active filament, right? There is breakage, right? And then simultaneously it relaxes and comes to the previous stage, that is the initial stage. So in this way, there occurs movement. Now I told you that the junction between this motor neuron and the sarcolemma of the muscle fiber, this forms neuromuscular junction. What I said? Junction between motor neuron and sarcolemma of muscle fiber. This is formed between motor neuron and sarcolemma of muscle fiber. Right? This junction is neuromuscular junction, and this is also called as motor end plate. Right? Now, here a neuronal signal, when it is reaching this junction, as I told you, this releases acetylcholine, or it generates the action potential in this sarcolemma, and this spreads through the whole muscle fiber, and then this causes the release of calcium from the sarcoplasmic reticulum into this sarcoplasm. Okay, calcium is released, right? Now this ACH or acetylcholine, this actually generates the action potential in sarcoplasm, which spreads through these muscle fibers, releasing the calcium ion that is present in the sarcoplasmic reticulum. So now what happens here? There is increased level of calcium ion. So this calcium actually binds with the troponin. Here this calcium, I had told you that this has troponin. This is a complex protein present over this tropomyosin at places, right? So first what happens here? This calcium ion, this binds with the troponin, and this removes the masking of that active site for myosin, right? Whatever the masking effect was there over this active site, this is removed due to the combination when this calcium ion combines with troponin, right? Now this energy which here is obtained is mainly by hydrolysis of the ATP. The myosin head now binds with the active or active site of actin, right? And this will form the cross bridge. So this pulls the attached actin filament towards the A band, right? Towards the A band or the myosin, over the myosin. Now, as you know, the Z lines, they are attached to the actin filaments. They are also pulled inwards. So there is shortening of sarcomere. That is contraction. Okay. Now, during this contraction, these I bands, they are shortened, but the A band, they retain their length. A is actually what? Due to the myosin filament, so it retains the length. Remember, but the I band, which is due to the length of this my actin filament, this shortens because this moves, right? Now the myosin now releases ADP and inorganic phosphate that you can see here, right? So here, after this energy is utilized, now this goes in the relaxed state. So that here again what will happen? A new ATP will bind, right? A new ATP will bind these cross bridges. It will be broken down, and this ATP is again hydrolyzed by the myosin head. Again, the cycle of the cross bridge formation will continue, right? So this process continues till the calcium ions are pumped back to this sarcoplasmic cisternae. That is resulting in the masking of these actin filaments, right? So this sarcoplasmic cisternae, that results in the masking of the actin filaments. So what I said, the process here will continue till the calcium ion is pumped back. It will combine with the troponin and remove the masking effect of this filament, right? Now this actually lead to the Z line to go to the relaxed state. Now repeated contraction of the muscle, this will lead to your accumulation of the lactic acid. Why? Because when there is excessive contraction of the muscle, there is anaerobic respiration or there is anaerobic breakdown of the glycogen muscle, right? So this will cause fatigue. This will cause fatigue, right? You must know that this muscle fibers, they also contain a red colored oxygen storing pigment that is known as myoglobin. So this is just like hemoglobin of the RBCs, right? So this is a oxygen storing pigment that is myoglobin, right? Here the muscles with high myoglobin content is known as red fiber. The muscle which has high myoglobin content, that is called your red fiber, right? And the muscle which this red fibers, they have more mitochondria also. Mitochondria are also more or maximum, right? So these muscle fibers, they have more mitochondria and more myoglobin, and they can use large amount of oxygen that is stored in them for the ATP production. Now these muscles, what will you call them? You will call them aerobic muscles. So what are aerobic muscles? They are the muscle fibers which contain more myoglobin, that is the red muscle fiber, and they have very high amount of mitochondria also, so that this can use the large amount of oxygen that is stored in them, right? Now, the muscle with less myoglobin and less mitochondria, they are pale in color and they are called white fibers, right? And these white fibers, as they have less number of myoglobin and mitochondria, but they have more sarcoplasmic reticulum. They have more sarcoplasmic reticulum. Remember this difference. They have more sarcoplasmic reticulum. So this can adapt the anaerobic source of energy for this respiration. They can adopt the anaerobic source for respiration also, right? Now next part I will take in the next class. Okay.